Right this very second, as you're sitting there, literally trillions of tiny little engines inside your body are performing this absolutely incredible feat. It's the process that keeps us all alive, it helps us heal, and it's how we grow. Today, we are going to unpack the amazing biological program known as the cell cycle.
I mean, seriously, have you ever thought about it? How does a paper cut just disappear? Or how do we all start as one single cell and become, well, us? The answer to all of that is the cell cycle. It's not random, it's this highly choreographed dance, a sequence of events with life or death precision. So why do our cells even bother? Why go through all this trouble? Well, cell division is absolutely essential for a few big reasons. For us to grow and develop, to renew and repair our tissues, like that annoying paper cut, and for some organisms, it's their entire method of reproduction. It's basically the foundation of life's continuity.
Okay, so before a cell can even think about dividing, it has to copy its most precious possession, its DNA. All of the DNA in a cell is called its genome. I want you to think of it like the complete instruction manual for building and running you. And a copy of this entire library exists in almost every single one of your cells.
Now, you can't just have this gigantic library of DNA floating around all messy. It's got to be organized, right? So it's packaged up neatly into these structures we call chromosomes. A typical human cell has 46 of them, and each one is just a supercoiled strand of DNA wrapped tightly around proteins to keep it compact and from getting all tangled up.
But here's the thing. Those neat, little X-shaped chromosomes you see in all the textbooks, they only look like that right when the cell is about to divide. Most of the time, the DNA is uncoiled and just loose inside the nucleus. As our source puts it, it looks like a bowl of spaghetti. You can't see individual chromosomes at all when it's like this.
And that brings us to a huge challenge for the cell. If one cell is going to become two new, identical cells, then each of those new cells needs a complete copy of that 46 chromosome library. That means the cell has to perfectly, and I mean perfectly, duplicate its entire genome before it divides.
Now here's a really key detail about those 46 chromosomes. They aren't 46 totally unique things. They actually come in pairs. You get one full set of 23 chromosomes from your mother, and a matching set of 23 from your father. That's why you have two of everything. 2 chromosome 1s, 2 chromosome 2s, and so on.
All right, this slide is super important because it clears up two terms that are so easy to mix up. So let's look at the left side first, homologous chromosomes. Think of these as the parental pair, like the chromosome 1 you got from your mom and the chromosome 1 you got from your dad. They have genes for the same traits, but they're not identical. Now look at the right. Sister chromatids. These only show up after the DNA gets copied. They are two perfect, identical duplicates of a single chromosome stuck together. So, the DNA is all copied. Now what? The cell is ready for the main event, mitosis. And this isn't some chaotic explosion. It is an elegant 4-step dance, and it's designed to perfectly separate all those copied chromosomes. But before we dive into the dance itself, take a look at this. It shows that a cell spends the huge majority of its life, we're talking about 90%, in a stage called interphase. That's all prep time. The actual division part, the mitotic phase, is just this tiny little sliver of the cell's entire life. An interphase itself has 3 parts. First up is G1, where the cell just grows and does its normal job. Then comes the most critical stage, the S phase. S stands for synthesis, and this is when all 46 chromosomes get duplicated. Finally, in G2, the cell does one last check and makes its final preps for the big split. Okay, now for the dance of mitosis itself. We can break it down into 4 key moves. First is packaging, where that bowl of spaghetti DNA tightens up into those visible X shapes we all recognize. Second is aligning, where all 46 of those chromosomes line up perfectly, single file right down the middle of the cell. Third is separating, where the 2 identical sister chromatids of each X are pulled apart to opposite sides. And finally, dividing, where the whole cell pinches in the middle and splits into 2 brand new identical cells. And the final result of this incredible performance? 2 new daughter cells that are genetically identical, not just to each other, but to the original parent cell that started it all. Each one has a complete and perfect set of 46 chromosomes. Mission accomplished.
So you have to wonder, how does the cell make sure this unbelievably complex process goes off without a single mistake? Well, it's not left to chance. The cell cycle has this sophisticated internal control system. A series of checkpoints that act like a quality control team to prevent total disaster.
The source material gave a fantastic analogy for this. I want you to picture the cell cycle as a spinning roulette wheel. As it spins, it keeps running into these little flaps that stop it cold. Those flaps, those are the checkpoints. The wheel literally cannot keep spinning until the cell gets a specific go-ahead signal.
There are 3 major checkpoints, and they each ask a really simple but critical question. The G1 checkpoint is like the main bouncer at the door. It asks, is it even a good idea for us to divide right now? The G2 checkpoint is the proofreader. After the DNA is copied, it asks, did we make this copy perfectly with no typos? And finally, the M checkpoint hits pause right in the middle of mitosis to ask, is every single chromosome lined up and ready to be pulled apart?
This control system is just so important. Because when those checkpoints break down, when the quality control team goes on vacation, cells can just forget the rules. They start dividing when they're not supposed to, and that can lead to catastrophic consequences.
And that brings us to the most important question of all. What happens when a cell's internal brakes just fail? When it barrels right through a red light at a checkpoint and just keeps going, ignoring every single signal to stop?
The result is simple, and it's terrifying. Uncontrolled cell division. The cell just loses its ability to regulate itself and starts replicating over and over and over again without any of the normal controls.
And this brings us to the ultimate why this all matters moment. Uncontrolled cell division, that failure of the cell cycle control system, is, in a word, cancer.
You see, at its very core, cancer is a genetic disease. It's caused by mutations, typos, in the very genes that are supposed to build the proteins for these checkpoints. So when those proteins don't work, those flaps on the roulette wheel fail, and the cell is just free to divide endlessly, which is what forms a tumor.
And this leaves us with a really profound final thought. Understanding the cell cycle isn't just some boring biology lesson. It's the absolute key to tackling one of humanity's greatest challenges. As the slide asks, if cancer is just our own cells forgetting the rules, then the future of medicine has to lie in finding even more clever ways to remind them.